So amino acids joined by peptide bonds. That is what we are going to look at now. So the chain of amino acids will give us a peptide or protein. To give us a peptide or protein. So two amino acids can react with loads of water molecules to form a covalent bond. So you see this one, this is amino acid, and this is also another amino acid. So this amino acid joined with another amino acid to form what we call a peptide bond with the release of water molecule. So you see in the process, a water molecule is being released, and this is what we call the peptide bond. This is the peptide bond. So one amino acid joined with another amino acid to produce a peptide. And the bond that involved is called peptide bond. So look at it. This one is amino acid and this one is amino acid. And there's a removal of molecules of water, and this is it. And we have our peptide bond, and this is the peptide bond. That is CONH. This is the peptide bond. So, in other way, we call it an amide linkage, and this amide linkage is planar. It's planar. So a pure double bond between carbon and oxygen will permit free rotation around the carbon nitrogen. So I hope it's understood. It's just that the most important thing that you need to understand here is one amino acid joined with another amino acid to do a peptide bond. And in the process of this peptide bond formation, a small molecules is usually produced or is usually released, being removed from the main compounds. So now we are going to look at the classification of amino acids. So classification of amino acids. So the classification of amino acid is based on the side chain. It's based on side chain. And this side chain classify the amino acid into non-polar amino acids, polar uncharged amino acids, acidic amino acid, basic amino acids. Then there is also other side chain structural classifications. You can also, they are classified based on the structural side chain. We have aromatic, we have cyclic, we have hydroxyl, we have thiol amino acids. So now we are going to start with non-polar side chain. We are starting with non-polar side chain. So in the case of glycine, the R side change is hydrogen. It's hydrogen. It means that this is how it looks like. For glycine, we have hydrogen here. We have COO. We have amino group. So the R side chain of glycine is hydrogen. So it means that we have hydrogen here. So we have hydrogen here. So it means that if you look at it, we have hydrogen here and we have hydrogen here. So in the case of glycine, it has two hydrogen attached to carbon. So that is why, if you could remember the last time, I also talked about that all the amino acids that have two groups, two distinct groups attached to the carbon, they are said not to undergo chirality, or they are not actually undergo stereoisomerism. So in the case of glycine, the R side change is hydrogen and already have hydrogen in the carbon. So therefore, glycine do not undergo stereoisomerism because of the hydrogen atoms. Then in the case of alanine, it's something, in the case of alanine, it's the something in the case of alanine, because in alanine, we have CH3 here in the other side chain. We have CH3, so it's alanine. So, and we should understand that the alanine, we have a ways of representing amino acids. We can use 3-letter notation and we can use 1-letter notation. So for the glycine is GLY. And the single letter is capital G. Then we have alanine. So for alanine is ALA, and capital letter A is used as a single letter for representation. Then for the valine, the R side change of valine is CH3 open bracket close to then CH, that is valine. It means that in the case of valine, this is what we have. In the case of valine, we have CH, then we have CH3. In brackets, we have 2. So all these, they are non-polar side chain. Why are they called non-polar side chain or non-polar amino acid? Is because they do not interact with water. Then we have tryptophan, we have phenylalanine. So it means that for the tryptophan, the R side chain is this phenyl ring. It's a phenyl ring that attached to this site. And then for the tryptophan, this is a ring that also attached to the R side chain. And for the proline, this is what we have. Isoleucine, this is the group that we have. The methionine, this is the R side chain. So all this, you just take them and attach them to this carbon because they are the R side chain. So all these amino acids, we call them non-polar side chain amino acids. So non-polar side chain amino acid include glycine, alanine, valine, leucine, phenylalanine, tryptophan, proline, isoleucine, and methionine. All of them are the non-polar amino acids. And this nature of this non-polar amino acid is predominant form at a pH of 7.0. So if the pH is changed, so there is a likelihood that the structure may change and they may not longer be considered as a non-polar. So it is at this pH that they are considered as a non-polar amino acids. And then the next thing is a polar side chain. So the polar side chain amino acid also, their predominant form is at a pH of 7.0. So we have asparagine, serine, glutamine, and we have threonine. So all these are also examples of polar side chain amino acid. When we said polar side chain amino acid, means that they can interact with water. And what makes them to be polar is because of the presence of OH group in the serine, the presence of OH group in threonine, the presence of carbonyl group and amino group, in glutamine, the presence of amino group and the carbonyl group of asparagine. And that is why they are non-polar. Sorry, that is why they are polar, meaning that they can interact with water molecule. Okay? So the next thing that we are going to look at is the acidic side chain or the acidic amino acid. So we have aspartic acids. Why is it that it is polar? Is because of the presence of this COO minus. This COO minus is what makes it to be polar. Look at it, this group. That is what makes it to be acidic. Also glutamic acid, this is what makes it to be acidic. And then also tyrosine. Then we have cysteine. So all of them, they are polar. Then we also have, they are acidic. Then we also have basic side chain. So for the basic side chain, we have, that is the basic amino acid. We have arginine due to the presence of this. We also have histidine. We also have lysine. So all these are basic amino acids. So you see, I told you actually from the previous class, from the previous slides, that these amino acids is actually classified based on their differences in terms of the R side chain. So I think, ladies and gentlemen, we are going to stop here for today's lecture. Then we will continue maybe on Wednesday. So you can ask questions if there is any.